All right, hello everyone. This is the exam review session for exam 3. So here we go. Let's just hop right into it. The first chapter on this exam is chapter 18. Oh, just as a reminder for this exam, make sure you bring a calculator because there will be some calculations. Okay, so chapter 18 here, there's not a lot that you guys need to focus on. So chapter 18 was mutations in DNA repair. So it's the big picture stuff here. So being able to know what the difference between a missense, a nonsense, and a silent mutation are will be important in understanding. So a missense is when it changes to another amino acid. A nonsense is when it's a stop codon, and a silent is when the amino acid does not change. So a nonsense mutation is most likely going to lead to what's called a loss of function and be a recessive trait. It's unknown what a missense mutation might end up doing to that final protein product. Then as we go down through this, we went into a lot of detail in that lecture about, so here it talks about if you forget what loss of function means versus gain of function, you can read over that and the different types of mutations that could exist. We won't have to do any calculations for mutations rate, but causes of mutations are important and what could happen that leads to that mutation. So of course there's spontaneous replication errors and spontaneous chemical changes that could occur. You don't have to know the specifics here. And then carcinogens that could also induce DNA damage. So a mutation is anything that changes the genetic sequence of a cell or an organism. Whether it occurs in somatic cells or the germline determines whether or not that mutation is actually heritable. So one of the worst kinds of mutations here are from ionizing radiation, X and gamma rays, which causes double-strand breaks. So double-strand breaks is when you have the full strand of DNA breaking in half, and that's a little bit harder to repair. So that high-energy radiation, that's why radiation is bad for you. So then we talked about transposons a bit. I didn't put anything on the exam directly for transposons, but DNA repair here is also important. For repair, just understand the different kinds of repair that exist. Again, I won't get really specific on each, but so something like a double-strand break repair from that UV light, you have to join those non-homologous ends back together. So understanding if you have a mismatch, you have to remove that nucleotide and then repair it. You have to do base excision, direct repair, mismatch repair. And so you want to fix it and get it back to the original sequence. All right, that is the DNA repair chapter. Next one here was population and evolutionary genetics. So for this one, let's make it a little bigger here. This one, remember, evolution is the change in a genetics in a group of organisms, not an individual, or not in any cell. It has to be in the germline too. So individuals don't evolve, populations do. So remember, mutations is that change in genetics. So primary mechanisms of evolution, these are just big picture things: genetic drift, migration, natural selection, and of course, mutations. Now, calculation-wise, this is where we will have a calculation on the exam. So the one I want you guys to focus on in this review session is this one down here in purple. We did this one in class as well. So this is looking at fitness and also using Hardy-Weinberg. So remember what Hardy-Weinberg is. It's p squared plus 2pq plus q squared is equal to 1. So if you have to refresh yourself on what p and q are, you can go up here and look at the previous info for how to calculate p and q. So let's say we have an example like this one here, where it's just talking about yellow peas and green peas, where green peas are the recessive version. So you want to know what's recessive and what's dominant. The dominant one can then come in two forms: big A, or big A, little a. Recessive one is always little a, little a. So if we tell you that the population is in Hardy-Weinberg equilibrium, that means it's not going to change. We have 27 homozygous yellow. So if they're yellow, it's big A, and a population of 382. So that's all the information you need to figure out P and Q in the frequencies of the different alleles. So here you could do the frequency of big A, and that's that 27 divided by 382. That gives you 0.071. So based on understanding the equation for Hardy-Weinberg, you know that is equal to P squared because A is represented as P and the recessive allele is represented as Q. So if you have two big A's, it would be P squared. So P squared is equal to 0.071. If you then take the square root of each of those sides, that gives you P. So make sure you can calculate P given information such as this. And then also, you know Q is equal to 1 minus P. So then you can calculate P and Q. Important to be able to do that. P and Q also represent the frequencies of just a single A, single big A, or a single little a. So the frequency of big A in the population is 0.266. The frequency of little a in this population is 0.734. So that tells you that little a is more of a favored characteristic. So then you can finish calculating what is the frequency of heterozygotes and the frequency of homozygous recessive using these equations here. So the frequency of heterozygotes is 2pq. So you need to get pq so you can use these equations. So 2 times p times q gives you this frequency. And again, I give you all these formulas on the exam that you will need. And then frequency of homozygous recessive is q squared. So you got p and q, you just plug and chug, and you get your frequencies right here. This one also then continued to ask what is the numbers in the population, but you don't have to worry about that directly. So then that also talks about chi-squared test, but I'm not making you guys do any chi-squared analysis on this one. So now those values can be further used down here for when we talk about fitness and natural selection. So understand this table right here for fitness. So if I give you the average number of offspring, so the fitness is equal to the number of offspring produced by an individual divided by the highest number of offspring. So here the highest number of offspring is 40, so you do 7 divided by 40, 8 divided by 40, and that gives you your fitness. And then your selection, or S, is determined by 1 minus W, which is your fitness. So then here we have 0.82, 0.8, and 0, because 1 minus 1 is 0. So you might have to fill in a similar table to that. It should be simple if you understand the equations here and how you do it. Now the next step is, so we know if we look at this population, the one that's being selected against are these dominant traits. So the recessive versions here, A2, which represents little a, these are the ones that are surviving and reproducing more according to this number 1 value. This 0.175 and 0.2 represent if an organism has that dominant allele, they're less likely to reproduce. So we can calculate what P and Q will be for the next generation based on W, based on the fitness. So we can do that here using these equations. So these are the important equations here. And then you will use that one later. So P nu is equal to P prime. That just is an easier way to write P nu. So this one is the frequency of A1 nu. So we have to calculate that. For that one, we use this. This is the frequency of A1 nu. So make sure you know how to use that equation. I show it down here as well. And it uses that same information as it uses the W1, 2 that we calculated right there. So here, this comes out to be 0.755. So we store that. That will be this term right there. But now we need the A1, A2, and that would be this equation right there. Again, I will give you these equations. So the frequency of A1, A2 nu, so this is the heterozygote version, is then 0.191. So make sure you can replicate this problem. So then you can calculate P prime. So 0.755 plus 0.5 times 0.191 equals 0.851. Check your answers when you do this. So if we know P is the recessive trait, how can we compare it to see how... so this is using the examples from above too. So where P initial is equal to 0.9, Q initial is equal to 0.1. And if we're looking at this fitness where P initial is 0.9, it now dropped to 0.85. So we're losing that P allele. And it makes sense. If you look at this, the P allele, which is the big A or A1 here, is being selected against. So it would make sense that that frequency drops and that the Q frequency, the Q nu, is going to increase. And this one increases from 0.1 to 0.149. So check your answers to make sure you did it right. Based on the fitness, you should get a rough idea of whether these P's or Q's should increase. So on the exam, you guys will need to be able to calculate P and Q, and then I'll give you some fitness information, and then you're going to show how P and Q changes in the next generation. So try this example, use different numbers in the example, and so forth. So it's a combination of this part right here I highlighted and this part down here I highlighted. So that's the important part from this section. I'm not doing questions really on the other parts here. So, but understand how genetics plays a role in evolution. And then here for speciation, understand the difference between allopatric and sympatric speciation. So allopatric is a geographic barrier, sympatric is no geographic barrier. And up here, for how we make a new species, it comes down to, are those zygotes isolated before fertilization, or are there problems after fertilization? So is it pre-zygotic or post-zygotic? And then those usually come down to whether those species separated via allopatric or sympatric. All right, so that's that chapter. And now we can move into bacterial control of gene expression. So we referred back to this a lot throughout the other chapters between bacteria and eukaryotes. Just remember this overall. So these are just big picture things of how you can increase or decrease expression, regulatory proteins, how they bind to something, produce a protein, and so forth. And then here, understanding how an operon works is important. If you can understand the 4 different types of operons, then you can understand how an operon works. So this is an important little table here, and I gave you guys handouts on this as well. So remember, if something is inducible, that means it's normally off because you're trying to induce expression. When a substrate is available, it is turned on. So if it's repressible, it's normally on. And if it's turned off, if enough product is made. So think about it when I ask the question and I say, when this is bound, the operon isn't producing the genes. When this other substrate appears, that protein is removed and is now inducible. So you're inducing expression of that operon. So then you can say whether it's positive or negative. So positive is when a regulatory gene encodes an activator and it binds. And then a negative one is when it encodes a repressor. So make sure you know these definitions, because then you can determine whether a question is saying this is negative inducible, positive inducible, negative repressible, positive repressible. So if it's negative inducible, you know the repressor is normally active. And so this is like the lac operon. So when a bacteria, when E. coli doesn't have the presence of lactose, the repressor is bound and you're not making those genes. When you have the inducer, so lactose is now present, that inactivates the repressor, and then you have expression. Whereas here for positive inducible, the activator is normally active, but not bound. It takes the substrate to then activate it. So here the operator is the activator. So an example of this one then could be something, what's an example? I didn't think of one beforehand here. So positive inducible is when you can have a regulatory gene is encoding the activator, and that means that regulatory gene is controlling whether or not that operon is being expressed or not. But something still needs to turn that gene on and off. So that's why it's inducible. So in the normal state, that gene is off. When a substrate is present, that gene is turned on, and the regulatory gene produces an activator. And then the other two then are negative repressible and positive repressible. So make sure you understand the differences between each of these. It all starts with, is that gene on or off from the start? That tells you whether it's inducible or repressible. So then, so we talked about the lack operon here, and so this is just big picture stuff. And we did that worksheet too. We did that operon problem. And we also did some examples in class too. So here, this operon problem, where the example was negative inducible. So, be able to repeat something like this. It might not be negative inducible, but be able to think your way through it and figure out, okay, what's the repressor? Is there a repressor? What's the substrate? What controls whether or not the gene is being expressed? And draw it out too. Draw it out too. That helps. And then think of whether there's a mutation. So what's it going to do in the wild type state if that substrate is present or not present? If you destroy the promoter, what's that going to do? If you delete the regulatory box that makes that repressor or activator, and so forth. So yeah, understand this and understand how we came to these decisions down here based on these mutations. And remember, if you forget what some of those are, I will tell you if like this little S1 down here, you won't have to remember exactly what the S meant. But the S means it's always active. So think how that will affect expression as well. So back to here then, so that's important. And then we also talked about trip operon. So that's just another example you can look at. That's also negative repressible. So there you can see the difference between negative repressible and negative inducible. So then, let's see what else do we have here. And that's all for that chapter. So the big thing in this chapter, I give you a situation and you can state, is that negative inducible, positive inducible, negative repressible, positive repressible, and then you understand how that changes things. Okay, so then we talked about eukaryotic control. Eukaryotics are more complex, so I wouldn't expect you guys to know as much from this. But the important parts here is how we do this regulation for what DNA methylation is. That decreases expression. So if you have an enzyme like methyltransferase and you have a mutation or inhibitor, you're not going to methylate and you're going to increase gene expression. Then you can also have acetylation of the histones. If you acetylate the histones, you're going to loosen the DNA around the histone proteins, and that increases expression. So understand what happens in acetylation and DNA methylation. Those are two important ones. So the table like this here, the two important ones are the methylation and acetylation. So this is methylation. I'll never give you a positive example. It's typically negative via methyltransferase. And acetylation is usually a positive effect on transcription. And remember, DNA methylation on the DNA, it only really occurs on one nucleotide, and that is cytosine. And it forms what's called 5-methylcytosine. And so that's where we come into these things called CpG islands, where those cytosines become methylated, and they stay methylated through rounds of transcription, so that gene regulation continues through cell division. So then here is when we get into a lot more detail that I'm not going to be asking you guys directly on this exam, between enhancers, insulators, all these silencers and so forth. It was just too much information to fit into this exam. So in here, also do know what RNAi is, and you should know that by now, and how we can turn off RNA or prevent translation via RNAi, especially when we start thinking about cancer as well. So yeah, that is that chapter. Now we can hop into epigenetics. And again, epigenetics has a lot to do with that methylation and acetylation again. And there we talk about the CpG islands again, and those being heritable. So a lot of this is just review of something we just talked about. And then some different epigenetic effects that could occur. They could be from the environment or from just your body determining it. And sometimes it's random, sometimes it's not. Epigenetics are very important in development and turning certain genes on and off at different stages of development. So we talked about just some examples of how epigenetics could have an effect. Important one here is genomic imprinting in the genome conflict hypothesis. So we talked about that IGF-2 gene, insulin growth factor 2, and how so genome conflict is when there's a difference between the father's sequence and the mother's sequence for genomic imprinting. They challenge each other. So the LGF2 gene from the dad makes the protein LGF2 or IGF2. I always say L. So insulin growth factor 2, IGF2. And then the mother imprinted gene controls the receptor gene. So if the receptor is there binding IGF2, there's a reduced offspring size. If the receptor is not there and the father's allele is making the IGF2 protein, then there's an increased offspring size. So if one of these is mutated, you can see how it could affect the offspring. So if this is mutated in the father and it's the recessive version, then you're going to get a runt and it's going to be a very small offspring. Or if this receptor gene in the mother is a recessive version, there's going to be an overproduction of the IGF2 protein from the father, and that's going to make a larger offspring. So you might have a question related to something like that as well. So the epigenetics chapter was really important because we referred back to it a lot. And now here in the biotechnology chapter, here is mostly big picture stuff. The bonus question, I'll tell you right now, is asking you some random ones of these and you define what they are. So if you go through this and you can say what each one of these techniques are and where they're useful, then you should do fine on that bonus question. But another important part here that might be its own question will be electrophoresis. So understand that the shortest fragments move the furthest, longest fragments move the shortest distance, and the direction of movement is this way, where the positive electrode is here, and the negative electrode is here, the positive electrode is down here. The DNA has a negative charge, so it moves towards the positive electrode. And in class, we did some handouts where we looked and compared the STR fragments as well. So remember, STR is short tandem repeats, and it's used in DNA fingerprinting. So if I were to give you a sequence for that, I don't think I have that worksheet in here. I don't. Let me see if I can pull it up real fast. Let me pull up that handout we did in class real fast so I can mention it. Handouts. Oh, great. Now it's not being happy. So I think it was this one here. So understanding the STR. So review this and remember what we're looking at here. So comparing person A, person B, crime scene. So read through this. This is an important part right here. So you're looking at the genetics between a mother, a child, and the father. So the child, so each of these lines right here represent a heterozygous version of each allele. So right here we're looking at 3 different alleles or gene locations. Remember, you have 2 versions of that gene, so different length of that gene. So here they're showing 2 different size fragments. If you're homozygous at a locus, then you'll have 1 line here. So the child then is a random assortment of the mother and the father. So this one, the child got this allele and got this one from the father. Down here, the child got this one from the mother and this one from the father. So the next child could have gotten this one from the father and this one from the mother. It's random. And then here, they both are, so this would be a key right here. So now that child is, we would know that child is homozygous for that particular trait because it got the same length fragment from the mother and the same length fragment from the father. So only one band right there. If the child was heterozygous, could have additional band right there or an additional band right there. So be able to read these and know that this is the positive electrode, this is the negative electrode, and these are the smallest fragments at the bottom. And yeah, that's that one. Genomics and proteomics, be able to define a genome, transcriptome, proteome, and epigenome here. So genome is all genes in a cell or organism. Transcriptome is all the messenger RNA, and the proteome is all the proteins. So which one of these is then the same in all somatic cells? That would be the genome. The genome should be the same sequence in all somatic cells unless there's a mutation that takes place. Transcriptome can vary from cell type to cell type, which then would cause the proteome to vary as well. Here is where we talk about the STRs. These are what are used in DNA fingerprinting and genotyping. Besides that, there's not a lot from this chapter. Mainly, I just wanted to give you an overview of genomics and proteomics. Then we get into developmental genetics here, which is the recent discussion from this week. Important parts from this chapter then is that it pretty much brings in everything we've talked about. So developmental genetics has a lot of epistasis basis in it, turning certain genes on, turning certain genes off. I won't ask specific questions on the Drosophila genes and the orders in which they're presented, because you can look up that information if you really need it. But understand that if a protein accumulates in a certain region, so anterior or posterior, so in a Drosophila, we have the bicoid and a nanos protein. Bicoid is anterior, nanos is posterior. So if nanos was put in the anterior end and bicoid was inhibited, you would have two posterior ends to that Drosophila larva. So these proteins are known as morphogens. They have a concentration gradient, and if that concentration gradient isn't right, you don't get the proper development of the head, thorax, and abdominal structures. So those would be the two I could ask about Drosophila, bicoid versus nanos. And how, if one ended up in the wrong spot, what would happen? And then I don't ask anything on these, and nothing directly on the development of males versus females as well. So you guys got lucky here that I didn't ask any direct questions on this. Right here, though, this is important. You might see a few questions on this. Teratogens. What is a teratogen? And what does it do? So it's an epistasis effect. It's not a mutation. You're not changing the DNA sequence. You're turning certain genes on and off that are important for development. So something like fetal alcohol syndrome. Or that could be caused by alcohol. Then you have drugs, radiation, infections, different things that can cause epigenetic changes and disrupt certain developmental processes in the body. So you might have some questions related to that as well. So then understand how, if it's related to something like this, how it could affect that development. So what is a teratogenic cause versus a mutagenic cause? And then how could that affect expression if I give you enough information? So mainly, understand what that is. It's a long story. And then we ended with cancer genetics. And in this one, the difference between benign and malignant is, of course, important. And And then also, what causes cancer? We know cancer is that uncontrolled cell division, but why? So there are different cell cycle checkpoints that are broken, and it's additional mutations are piled on that then leads to that cell to be able to become malignant and metastasize and grow tumors other places in the body. So you have cytoskeletal issues that pop up and so forth that allows that cell to move around and get away from the normal cell cycle checkpoints. So remember, cell cycle, G1, S phase, G2, and so forth. And for this one, again, it's hard to get really, really specific because we got specific in class. So I won't get that specific on the exam. If you guys did the quiz for this one and all the previous chapters, I used a lot of similar questions from the quizzes on this exam for the multiple choice. So there are 28 points of multiple choice, and they're each worth 2 points. So make sure you understand the big part here is what is a proto-oncogene, what is a tumor suppressor gene, and how a proto-oncogene, when it becomes an oncogene, is usually a dominant mutation, and a tumor suppressor gene, when you turn that off, is usually a recessive mutation. It's a down regulation, and an oncogene is an up regulation and a gain of function. If you understand that difference, then that will help a lot. Because I will give enough information for you to answer the question based on whether telling you it's a tumor suppressor gene that is now inactive or an oncogene that is now active. So if you have a tumor that's growing and you find a tumor suppressor gene in it, you'd expect it to be downregulated with a loss of function. And that could be from higher levels of methylation. Maybe for some reason that gene was methylated and now it doesn't work as well. So different mutations here. Again, it's all about the gain of function for oncogenes and a loss of function for tumor suppressor genes. And then, like I said, this could be due to epigenetic changes like methylation or hypermethylation, expression of interference RNA, or loss of interference RNA. And then how a virus might promote cancer production because it's carrying that proto-oncogene and increases the chance of expression and mutation into an oncogene. So yeah, that was everything from that. Let me scroll through real fast just to make sure I hit on just about everything. And I think I did. Like I said, the big one is going to 15 points for doing the population genetics problem in terms of fitness. So be able to do those calculations, understanding mutation types, and then the epigenetics and the different causes for cancer, and operons. Of course, you have to understand the operons and so forth. So besides that, I feel like everyone should do very, very great on this exam. So study the quizzes and study the important parts I've focused on in this video, and you should be great. All right, everyone, have a great day and good luck.